Welcome to AP Biology. Today's presentation is going to be on how cells activate cell division in cancer. So, cells activate cell division by getting cell signals. The communication can come from internally. When cells are getting large enough, they will signal to themselves that it's time to divide. They could come externally. For example, if I scrape my knee, cells will signal near the scrape that it's time to divide and repair the damage that was done. All of these signals, though, come from what are called growth factors. What are growth factors? Well, here's an example of a growth factor. This is called platelet-derived growth factor. This happens to be dispersed when we have an injury. And it's made by platelets that form in the blood clots. And it gets released to the surrounding cells around the scab, and it tells those cells to start to divide. Here's a little experiment they did. They took some connective tissue and chopped it up and put it into a suspension and then plated it out. And if you add growth factor to the growth medium, you get lots of cell division, and if you don't get any cell division. And so it's pretty obvious that this has a major effect on cell growth. And the way it works is like this. Growth factor will bind to a receptor. This particular receptor is called receptor tyrosine kinase, and it will bind to this receptor and it will activate a chain reaction where molecules are causing a phosphorylation cascade. SOS, RAS, RAF, MAC, ERK, these things carry a signal basically from the cell's surface to the nucleus to tell the nucleus it's time to divide up our chromosomes. So we have to replicate the chromosomes so that we can divide them. There are many molecules that are involved in this whole process. Many of them are what we call cyclins and CDKs. These are cyclin-dependent kinases. Kinases are things that are going to phosphorylate other molecules. And so when we activate a kinase, a lot of times these things will be involved in phosphorylation cascades. These cyclin-dependent kinase, cyclin complexes are going to trigger the process to go from one stage of the cell cycle to the next. So basically when you're going from G1 to S, you get a big spike in a certain type of cyclin, which is going to push the cell into the S phase. And then you get another big spike of S cyclins during the S phase. And then you get another big spike of cyclins that pushes the cell into mitosis when you get to the end of G2. So these molecules are around in various amounts kind of throughout the cell cycle, and they help the cell move from one stage to the next.